Okay. So we'll wrap up the part about G-protein carbon receptor by the end, by the questions that you're supposed to answer for today, and then we'll talk a little bit about what is the assignment that you need to do for the course, and then we'll continue with post-transcriptional gene regulation. And today we're supposed to be 3 hours, so you need to be strong. But also I have a little bit sore throat, so you'll have to ask a lot of questions and long questions that have short answers also. Okay. So, you know what, let's start from here, and we'll go like this. And if you don't know it, it's okay. Don't. Hi. When signaling molecules are received by the same cell that secreted them, the signaling is called autocrine, and this you can answer even without knowing what signaling is, because it's auto. Yeah, we talked about the 3 different types of signaling. If it's endocrine, that is released, that is signaling that affects, that is affected by molecules that travel long distances. Paracrine is from close proximity, like the connection between cells. For example, synapses can transmit paracrine signaling. And the autocrine is when the cell releases a signal or releases a molecule that is recepted by the same cell that was that released the molecule in the first place. So, which sentences are incorrect about G proteins? Yeah, stop. Which of these sentences are incorrect about G proteins? Is all G proteins activate their target effector proteins? Is it true? You can guess. If you have all here, and it's biology. Okay, great. So, in general, nothing in biology is all, and I think in life in general, but so not all G proteins activate their target effector proteins. Some of them inactivate the target effector proteins, like we saw in the example of adenylyl class, that a certain type of G protein deactivates the enzyme, and another type of G protein activates. So, the alpha subunit does not always facilitate the downstream effect. Is this correct? You have here not always. Okay, so yes. So I'm trying to give you like tips about how to answer multiple choice. This is how whoever studied biology, this is how you do for most of the tests. You know, you just go, you understand how the question is formed, and that's how you answer. So the point about this is the alpha subunit is not always the subunit that facilitates the downstream effect. Sometimes we saw examples of the beta-gamma subunit, okay, for like in the example of the muscarinic receptor and the opening of the potassium channel. And in that case, we have the beta-gamma subunit. The trimeric protein is transmembranal. Is this correct? What is a transmembranal protein? Yeah. So when we're talking about trimeric protein, and I mean G proteins. Okay, G proteins are, we normally learn about trimeric G proteins. So are there transmembranal? No, they're just anchored to the membrane. And the alpha subunit is capable of GTP hydrolysis. Is this correct? What?
Doesn't it need help from another unit?
The question is not if it needs like help, but if it has a capability to hydrolyze. And actually the GTPase is located in the alpha subunit. Okay? It's right that just after you have the activation of the G protein coupled receptor, then it allows a conformational change that permits this hydrolysis, but in general the hydrolysis is happening in the alpha subunit of the G protein. And the disassembly of the G protein requires GTP hydrolysis? Yes. Okay. So, yeah, the disassembly of the G alpha protein from the G beta G beta gamma subunit is facilitated by GTP hydrolysis. Okay. Next question.
To conclude, A and C are linked to
the all G protein activated. Yeah, A and C. Yeah. A and C. Okay. So below are different steps in signal transduction pathway of G protein via the analytic class, which we learned about both in the article and the class. Arrange the steps in the order of occurrence. So, what is the first?
the G protein activation.
Right, and after the G protein activation?
It activates the analytic class, which activates, which increases count level,
Mm-hmm.
which V2 count activates pKa, so that causes pKa activation, which then correlates targets in the stack, which
Right. What is he doing? If I press this, great. Okay. You're correct. Is this clear for everybody? This sequence, because it's important. G protein coupled receptor signal transduction response is regulated by. So.
I wasn't sure about B, but completed all of the others.
Changes in local concentration of protein components. Where did we see this? Do you remember? Where did you see with this example? Right, in the visual system, when we talked about the rhodopsin cells, about the rod cells, we saw that the G proteins move to different cellular components when they're exposed to light. So changes in affinity between the G alpha and G beta gamma subunits. It's correct that we didn't see any examples of this happening. Phosphorylation of the transmembrane domain of the G protein coupled receptor. In what, can you remember in what respect did we talk about that? About phosphorylation?
The transmembrane or like the cytosolic membrane?
Okay, right, yeah. So actually, we talked about phosphorylation of the cytosolic domain of the GPCR, not the transmembranal domain is normally not highly approachable. Not very approachable for proteins or kinases to do phosphorylation. And endocytosis of the GPCR. What is this like? In which responses did we see this? Like, when will endocytosis happen? Do you know? You have a guess? After you have, we talked about that after we have massive activation, okay, of the G protein, and we have massive activation of the protein kinases, and because we have all this phosphorylation that happens to the receptor itself, then it reaches to a point when an arrestin molecule binds, and the binding of the arrestin molecule can cause endocytosis of the G protein coupled receptor itself, okay, and that's is inducing a regulation, or it's like a negative feedback of this response. So it's, I always forget to do the conclusions, but yeah, it's A and D, because these two are not mechanisms that we talked about. It also if they happen, they're not common. So, last question. The arrestin protein. What? Which one of these? This binds to G protein and prevents its activation by a receptor. That's the arrestin molecule. We saw it doesn't bind to the G protein itself. The G protein is the unit that moves along the membrane, right? The arrestin actually binds to the receptor, okay, through phosphorylation sites and prevents the activation of the G protein. Okay, so all of these are incorrect, and B is the correct. So you need to, I hope you will refresh and you remember a little bit before the test, because this is the kind of questions, again, like I told you, the kind of questions that we have, and a lot of these questions are actually like if you know it or not know it. So I know it's a lot of memorizing, but in the ICNC there's not a lot of memorizing in general in the courses. A lot of so it's good that you will have. You need to look at it as like expanding your biological vocabulary. Okay, so memorizing may seem dumb, but it's important to know the different factors, and when you will read articles in the continuation of your rich scientific career, then you will be able to understand what you're reading. Okay, so where is this? The final assignment. I will publish this, just a minute, you're not seeing it. Okay, so I will publish this presentation obviously online. And the idea of the final assignment is 30% of the grade, okay, of your final grade, is to give you motivation to do a good job. And this is like a programming code, how you're supposed to select your research paper. So you need to choose a paper, search online. I recommend Google Scholar to look. So you can type impact factor for the journal that you found. If you write like impact factor. Yeah, it has to be. An impact factor is a factor in the journal. Yeah. So I'll talk about impact factor, which is very important. Impact factor is like a general. It's like a way, maybe not a very good way, but it's still the most accepted way to determine the importance of a specific journal. Okay, so for number of citations. It's a derivative of the number of citations, but not only that. And but it's called impact because it's like the propagation also of citations and secondary citations and stuff like that. But I actually don't know how it's calculated exactly. But for example, if you look, normally the journals go like in three categories. You have the high impact. High impact are the ones that are above 10, and in these journals you will find all the Science, Neuron, Cell, all the journals that have one name, okay? Normally except PNAS. So PNAS I think today is like 13 or something like that. But again, if you find like something that is 8, it's also okay. Doesn't have to be like we don't have to be very, very strict about that. The idea is also because it's not that you don't find good articles that are published in low impact journals, but one of it does show you something about the interest and the strength of the results that are published in that journal. Okay. Again, it also really depends on the field of research. For example, when you're talking about computational neuroscience, because it's relatively a niche, so 5, like if you have a journal for computational neuroscience that has impact factor 5, so it's high. Okay, so but in biology or in general sciences, then you can find journals like Nature and Science, or like 33 or something like that. But it doesn't always mean. For example, like journals of medical journals can have an impact factor of 50, just because they have a lot of impact. Okay, so after you select a high impact factor paper, it has to have a molecular and neuronal component, and I will elaborate about that shortly. Then you need to read the paper, even briefly. I suggest to really look into when you want to scan papers, start from the abstract. Okay, like read thoroughly the abstract, and then look at the figures of this is like the order that I normally do like research or journal scanning. So read the figures, see if it's interesting, if it has also a neuronal and a molecular component. Then send me the paper for approval. If I said yes, then you can proceed to the next step. If no, you need to return here and do this all over again, but don't do this too long. Okay, so I will talk about that now. But before that, these are obviously we'll have these as guiding questions, but you have to write an essay about that journal. It has to be, the requirements of the essay is that it won't be very long. Okay, so maximum is 3 pages, minimum is 1 page, but it can also be like equal to 1 page. Font 11, spacing 1.15, normal margins, font, format, and the name of the file. Okay, and the deadline, is this the deadline? No, probably this is not the deadline. This is before the course. Okay, so maybe it's the opposite. I think it's the opposite. It's supposed to be the 1st of February, so it's like after your exam period, right? Or in the middle? Okay, so anyway, there's not I need to see when I need to submit the grades, and before that date, I don't mind if you submit it whenever you want. And don't choose a review paper, okay? I wrote this here. A review paper is not really, it's not the point. It describes too many research questions and too many researchers, and it doesn't go into the depth of the research itself. So, and the questions that you need to ask are, what was previously known about the neuronal molecular elements discussed in the paper? What was the main finding in the article? Did the researchers prove their point? Okay, so remember when we talked about
opinion article, okay?
What?
An opinion article.
In general, we need to have a research article. Okay, so it needs to be a research that something was already known, like about the question. They added something to this question, then they proved their point, like I said in the past. You need to suggest a follow-up experiment and a molecular experiment that will help support the findings, or maybe it can be like any type of follow-up experiment. And in the end, why is this an interesting article? Okay, and I wrote be intelligent, because don't write like it's interesting because I don't know, like because I like it or something like that. It's interesting because the color of the font is really nice. So try to see something in like a wider context and explain why it's interesting and why it's important, like in that sense. So a molecular element can be any type of cellular process that we learned in class, such as RNA splicing, protein folding, transport of ions, for example, or molecules, genes, coding or non-coding. Like especially now we're going to learn about genes that are non-coding for protein, signal transduction element, proteins are involved in proteins are involved in signal transduction, ion channels, ATP pumps, etc. Okay, so it has to deal with something with these components. It's not enough that they just like there are a lot of articles in neuroscience that they just like took a virus, infected the cell, and overexpressed. Actually, if you focus, it depends. So in that sense, it really depends like how they treated the molecular element. Okay, did they actually describe it? Did they develop it? So that's very important. That they just didn't use. If they just used the method that they took from somewhere else, then it's less interesting. But again, if you have questions, then feel free to ask. A neuronal element can be any cellular process that involves electrical activity, neuronal functions, synaptic plasticity like LTP or FTD, the morphology of the cell, like of the neuron, if we're talking about the synapse, or dendrite, or axon, or whatever. It can, it can also be glia. Yeah, it can also be glia. It can be, and even if we're talking about like cognitive processes, behavior, or neurological, or related to neurological diseases. Okay? So try to find articles that fit into these parameters, but again, I'm sure there are a lot of things here that I missed. So just ask me, and I'm sure it'll be okay. Questions? Okay.
So there was no presentation of
No. Like I said before, we're trying to we don't want to torture you so much. So like if we would have to do presentations, then we'd have to have much more classes, but we don't have time for this presentations. And also this task, if you really sit on it seriously, I think it's not supposed to take you. You can basically finish it in a day. Okay. So the idea here is not to give you such a hard assignment, it's just to give you a chance to study a paper and get a feedback on that. So that's what's important about here. Okay. So last time we finished here, and I described the mechanism of the spliceosome, okay? The different components or different proteins that are in charge in breaking apart introns and or removing introns and attaching exons. And I said that this complex is composed of about 70 proteins, and these proteins are working together with small RNA molecules that facilitate the binding of between the mRNAs and the protein that are acting here, and that the elongation step of transcription is coupled to the presence of RNA processing factor, meaning that this process a lot of times happens while the mRNA is getting transcribed. Okay, from the DNA, when the mRNA is formed, many times the splicing or the factors that are associated with splicing will start associating and binding to the growing mRNA. Okay, so one of the things that I told you, and I explained this, and we saw it here, that the actual sequences or the motifs that tell the cell that this is an intron, that this part is an intron, and this is an exon, are relatively not very stringent, or not very not very conservative sequences. You just have two nucleotides that you have to have in the beginning, which is GU of the intron. Then you have to have AG and an A somewhere along this from 20 to 50 base pairs or bases from the 3' splice site. And then you have a region that is rich in pyrimidine nucleotides. So in general, you can have in random, you can have this sequence, or you have a lot of times that you'll find like a GU sequence randomly across RNA molecule. Okay, so you have to have some other elements that will tell the cell what is an exon and what is an intron, and these elements are normally called SR proteins. These proteins bind to to recognition elements or to recognition sequences that are normally enriched in exons. Okay, and this whole complex over here, including the proteins that recognize, for example, the branch point of the intron, or pyrimidines or the AG here, together with the protein that recognizes the GU of the next intron, together with these SR proteins, form this complex, like this huge complex that in a sense can tell the cell this is an exon. Okay, so then the identification of this whole complex identifies what is an exon, and the spliceosome.
Can you repeat again what the SRs are?
The SRs are proteins that know how to bind to specific elements that are rich in exons, okay, that are normally found in exons. And because they bind to these elements, they actually define where an exon is. Okay, so they mark the exon. Okay. So, but actually, when you think about it, it's pretty crazy because you have to have because these exons. What are these exons? They're coding for proteins, right? So these elements, their sequence has to have also a reading frame, meaning like triplicates of codons that will code for amino acids and will code for proteins. But also inside them, they have to have like a secondary code on top of the first code that will have elements that these SR proteins will know how to bind to. So the complexity here is pretty high. And what we define as a spliceosome is this unit, or all the proteins or all the elements that are associating to the RNA or to the intron between the SR proteins. Okay, so this is the spliceosome that we saw here. Like all these elements compose the spliceosome. This huge machinery that causes this whole thing to occur. Here, so I won't forget that some introns are self-splicing. Okay, and what do I mean self-splicing? When we look at the lower life forms, such as fungus, we can see sometimes that you have introns that actually splice themselves out. Okay, so they don't need almost any help from other proteins or from like a spliceosome or something like that, and they can, you can say, like spontaneously splice themselves. And why is this important? Not because like in humans or higher vertebrates normally it doesn't happen, but the fact that this can happen is important because, like I told you before, that this splicing or the breaking apart of an intron and the connection between two exons does not require energy. Okay, so we describe this reaction as a trans-esterification reaction, that actually doesn't, in this cycle, there is no requirement of ATP. Okay, so, and again, this is, if like as a scientist, when you look at something like that and you say, I have here like a random process that can occur, like RNA editing that can occur without any ATP, so it may indicate that this process is very, very ancient. Okay, like maybe before you had ATP, before you had any like complex molecular element, this could happen like in a spontaneous fashion. This? Yeah.
Is this binding of all these proteins is something that
first one to bind and then everything binds after? I mean, I don't understand.
Well, it doesn't happen in parallel. You have like first the element of the U1 and U2 that bind to these elements, and then they recruit the other elements, and then they recruit other elements, and then the conformational change. And this whole process is very like complex. But and then you have like after all these proteins are bound, then you have like this huge conformational change that almost includes all the proteins there. It's like I told you in the example of the Megazord, that everything changes and it turns into something else that actually facilitates the splicing. But did you mean like in different introns? So it doesn't happen like one intron at a time. It happens in all introns. It can happen like simultaneously in all the introns.
And the SMRs aren't part of the splicing.
And they're not defined. They're not defined as part of the splicing.
They come there and then the splicing knows like where to look.
They come there and through the interaction between the SR protein and the U proteins, which are part of the spliceosome, then this whole process happens. But you're right that every time that we focus on a specific function, then all the other functions are like, or the other proteins are like ignored. Okay, so for example, here you'll find like this naked loop of RNA, or here this mRNA again is naked. And like I told you, this is not the case. Normally, mRNA is covered with RNA binding proteins and association proteins, and also other small RNA molecules. Okay, that facilitate this association. But in general, what we define as a spliceosome are these components over here. These are the main components that define the spliceosome. But each one of them is composed from many, many types of proteins and small RNAs.
But the SR are proteins, not small RNAs. That's what I meant by SR. Like the ones in the next slide.
Yeah. SR proteins.
So, and they do come there before the spliceosome, or?
I don't know how to tell you if the U2 or U1 like come before the SR, but I can guess that in general maybe the SRs are one of the first that are binding. But a lot of time you'll see when we talk about alternative splicing, then you'll see that sometimes like maybe the kinetics of the binding is what determines whether a certain exon will be included or excluded, and a lot of these things are determined by the dynamics of the process itself in the cell, and again, a lot of this is unknown. So a lot of
Okay, and when you said that some exons can splice themselves, like in primitive animals, so that's with SRs or without SRs? Like
so.
You said it's without spliceosome.
Actually, the SR proteins, so these, in these lower, and I said again, lower life forms, they're not animals, they're fungus. Okay, so it's not, normally in animals you don't have self-splicing. Then there are other elements that resemble elements that associate to the SR proteins and resemble elements that associate to the U1 protein, but they're just like small molecules of RNA that actually function, and this whole that process is not mediated by other proteins. Okay, so that's like that. It doesn't mean that there's nothing else there. Okay, it just means that there is no spliceosome, or there's no huge protein complex that facilitates the splicing. Okay, so when we're talking about splicing, because we have different exons, one of the most common features, especially, and now I'm talking about higher organisms, and you can there's a debate about if a human is like higher life form, but in general we see that in higher organisms we have a lot of what we call alternative splicing, and alternative splicing is a process of exclusion of introns and reattaching of exons in different combinations. Okay? So for example, if this is our gene on the DNA, and it has 5 exons, this is how the pre-mRNA molecule look like, and this molecule can yield, or can yield, in this example, 3 types of different mature mRNA molecules, and thus 3 types of proteins. Okay, so you can have either you can have all the exons together, or in this case we're missing the 3rd exon, or in this case we're missing the 4th exon. Okay, so there are several processes that, again, like I told you, in each gene it can be something different, and a lot of these processes that cause alternative splicing are unknown. But we know that mRNA variants are that if we're looking like in the same type of cell, in the same tissue, a lot of times you can find in different types of cells different types of variants, and also in the same type of cell you can also find different variants of the same protein. Okay, so this alternative splicing, and it's estimated that in the human genome 75% of all the genes are alternatively spliced. Okay, so this is an enormous number, and it significantly elevates the level of the complexity of the different types of proteins that we can have. And if you remember when we talked about in the beginning of the semester about modifications that can happen to proteins, so just think about all the possibilities that you can have of different proteins, and this is just arising from one gene. Meaning that if we're looking at the 22,000 genes that we have in our genome, for 75% of them, that it has been shown that from the same gene they can have at least two different variants. Okay. So it doesn't mean that actually 95% or 99% of our genes undergo splicing. Okay, but not all of them go alternative splicing. So an extreme example is from the DSCAM gene, which is abbreviated for Down syndrome cell adhesion molecule. It's actually discovered and expressed in the retina of the Drosophila, but it's also expressed during the development of the brain. And for example, the DSCAM gene has 95 alternatively spliced exons, yielding 38,000 possible isoforms. Okay, so a single gene has more options to create different proteins than our whole genome. Okay, so and actually this protein is located in chromosome 21, and this is how you this is like a map showing areas of chromosome 21, and it's actually in Down syndrome this chromosome has 3 copies. Okay, and this is what we call trisomy, and thus this gene is also overexpressed because it has more copies, and it's still unknown what exactly is the function of this gene in the Down syndrome. But because this gene is responsible, because it's a cell adhesion molecule, and it's responsible for interaction between 2 cells or between 2 neurons, and the recognition between 2 neurons. Because when you think about it, I guess that you ponder about the neural system or nervous system that during development, how does each neuron know which neuron it should connect to, right? So it's a very, very complex structure and very complex mechanism. And one of the factors that determines if a neuron if 2 neurons will have a synapse between them are the adhesion molecules that the cells express. And one of these adhesion molecules is this molecule, and then it's easy to understand or to guess why it has so many types of isoforms. Okay, so each one of these isoforms can mean like a different signal for a cell that is seeing this protein. Question?
Can you repeat what cell adhesion molecule is?
A cell adhesion molecule normally is a molecule that facilitates the connection between 2 cells. So if you have a membrane, there's a double membrane of one cell, there's a double membrane of the second cell, so you can have a protein that looks like something like this, and the other will be like something like this, and this element can be like a cell adhesion or scaffold protein that connects like the cell. It doesn't have to be for a different cell. It can be also for the extracellular matrix. You don't always have like 2 adhesion molecules. It can be like this cell is connecting to like a collagen chain or something like that is extracellular or something like that. So in general, cell adhesion molecule just indicates that this gene is involved in the attachment of the cell, can be to another cell or to another factor that is extracellular. Okay. One of the nicest examples again, we talked about, last class we talked about the visual system, now we're talking about the auditory system of alternative splicing happens in hair cells. And you didn't learn about the auditory system here, right? I think you're going to learn about it in physiology. But in general, we have this special organ, or the basilar membrane, that is inside our inner ear. And along this organ, you have vibration sensing hair cells that can feel the vibrations coming from this membrane. And if they're in one side of the membrane, they will be sensitive to, like, for example, 50 hertz oscillations, and here they will be sensitive to like 5,000 hertz oscillations. So like it's a topical organization along the axis of this membrane, and because of the physical properties of this membrane, it will vibrate. Like if it receives or hears a frequency, so each frequency will elicit vibrations in different sections of this membrane. Okay, and this is one of the reasons that we can identify like different frequencies and tones. But above this, the effect, the mechanical effect of this membrane, cell sensitivity can also be determined or achieved by alternative splicing, meaning that different splicing variants of the gene SLOW, which is a calcium-activated potassium channel that is responsible for the oscillation of the memory potential of these cells, has a tendency, if the different splice variants or the different splice isoforms have different thresholds of opening to different concentration of calcium. Okay, so and not only that each hair cell expresses specific combination of these isoforms, and thus the alternative splicing actually helps to do like tuning, like further tuning of which of the frequencies this cell or this cell will be sensitive to. So, addition to the physical properties, there are also more properties that determine like also like there's neuromodulation, and there's adaptation, and there's like the changes in the size. It's actually really nice. You know what? I will show you. I thought I would not do that, but because we're in the end of this first class and we're in a long class. So another method of adaptation or changing the gain of the response of the hair cell comes from, and it's not related to what we're talking about now, but it comes from that the hair cell have the ability to change their shape, okay, in a very, very rapid manner. And the changing of the shape actually changes the concentration gradient, like, or the between different areas of the cell. So if this is like the hair cell, it can be either in this state, or if it hears a specific frequency or needs to like amplify the frequency in a mechanism that we're not going to talk about, it can undergo like a morphological change and become like fatter and lower. Okay? And the reason that I'm telling you that is just because there's a really nice video which they isolated in a hair cell, and they played for the cell music. Okay, so they saw how it changes, how the cell changes its morphology in response in response to that music. Did you ever see this video? Okay. I want to see if it works.
1, 2, 3 o'clock, 4 o'clock, rock. 5, 6, 7 o'clock, 8 o'clock, rock. 9, 10, 11 o'clock, 12 o'clock, rock, we're gonna rock. Around 7 o'clock tonight, coast, back,
Okay, and this has nothing to do with alternative splicing, but it's also cool. Okay, so we'll take a break, and we'll continue next class. יש אנשים שאומרים שזה בכלל לא משהו בשיר הזה בכלל ש... זה יסתדר יפה מדי. זה יסתדר יפה מדי. סליחה שהייתי בעיקרון של לדעת איזה תדר שמענו. בנוסף לזה גם עובר אוסילציה, ולא רק האזור. זה הממברנה הבזאלית שכיוונה מלמעלה, זה יכול לעבור, אז פה היא תעבור אוסילציה, כאילו בתדר כזה, ופה היא תעבור אוסילציה בתדר כזה. לפי האוסילציות האלה, גם תאי שערה, עוברים אוסילציה של המתח ממברנה שלנו. ולפי האוסילציה הזאת, זה בעצם, זה מחדד יותר את לפי היכולת שלה לעבור אוסילציה של פרק מסוים, זה מחדד יותר על איזה תדר הם יהיו מכוונים.
נכון.
why are you desperate?
לא, אבל מה שקובע את כי זה שהאוסילציה שלה, זה שהממברנה תעבור, זה שהממברנה עוברת אוסילציה, זה לא אומר שאחרי זה ההשפעות של זה יהיו...
אז מה באמת, הממברנה מה היא משנה שמשפיעה על השערות?
הממברנה מפעילה פותחת תעלות שמשפיעות על הפוטנציאל של הממברנה, ואז בתא עצמו יש שינויים נוספים שקורים שבעצם מגבירים את הסיגנלים של האוסילציה.
כאילו השערות אבל הן מחוברות לממברנה, כאילו הן בעיה מוטציה?
זה ממש משהו פיזי כאילו שמשנה את אנחנו לא נסתכלים על זה יותר מדי, אבל בעיקרון יש פה מין חיבורים מיוחדים, חיבורים מיוחדים שמחברים את זה, פשוט בצורה מכנית. ברגע שיש תנועה, זה פותח תעלות. אוקיי? והאוסילציות האלה אחרי זה צריכות להשפיע האוסילציה של כל המתח בממברנה. והקדיעה שבעצם
אני לא הבנתי איזה תעלות נפתחות. כאילו, אני רואה הממברנה זזה.
כן, כי אנחנו לא נכנסנו יותר מדי לעומק. זה הממברנה. והיא זזה בצורה כזאת. אוקיי? למעלה ולמטה. כשהיא זזה ככה, השערות בעצם מסודרות בצורה כזאת. אוקיי?
אוקיי.
וכשהיא זזה היא לוחצת בעצם על השערות האלה.
אוקיי.
והתנועה הזאת גורמת לחלבונים מיוחדים שהם כמו חבלים.
אז השערות הן בעצם מתחת לממברנה.
כן. השערות מתחת לממברנה.
אוקיי, סבבה.
והתנועה הזאת גורמת לפתיחה של תעלות שמכניסות איונים בעצם.
אוקיי, סבבה.
עכשיו...
איזה איונים הם מכניסים? זה הכנסת...
הם מכניסים איון של סידה. אני חושב שזה איון של סידה. והאיון של סידה שנכנסים בעצם בעזרת התעלה הזאת, בעזרת הפלואו, זאת אומרת שפותחים את התעלות פוטסיום,
שלאן הן משדרות, שהן כבר...
שהן משפיעות בעצם על המתח ממברנה עצמו, כי משנה את כל המתח ממברנה של התא.
איך הולך הפידבק לופ הזה?
אז יש איזשהו את התעלות האלה, וברגע שהתעלות האלה נגיד חשות, אם יש עלייה ברמה של הקלציום, אז התעלות האלה יכולות להיפתח. אוקיי? עכשיו, כל תעלה תיפתח ברמה אחרת של קלציום. זאת אומרת, אתה צריך, אם נגיד תעלה...
אבל לאן הן משדרות? כאילו באז עובר שם קייב ולאן הוא...
ואז לפי הדפולריזציה, השינוי בדפולריזציה הכללי של הממברנה יגרום לשחרור של נורוטרנסמיטר לתא שיקלוט את ה...
זה הממברנה הזו ולא הממברנה למעלה.
כן.
לא הבנתי.
כן. יש את הבאזלר ממברנה, זה איבר גדול, ויש את האוסילציות של הממברנה של התא עצמו.
למה שחרור של אשלגן יגרום לאוסילציות בממברנה של התא?
כי לא שחרור של אשלגן, אלא פתיחה של תאות אשלגן.
כאילו פתיחה של תאות אשלגן יגרום לשחרור באשלגן.
פתיחה של תאות אשלגן יגרום ליציאת אשלגן כאילו מהתא, האמת שבמקרה הזה זה כניסת אשלגן. והכניסת אשלגן זה מה שיגרום לשינוי של הפוטנציאל, כי יש יותר עניינים חיוביים בפנים. אז הפוטנציאל של הממברנה ישתנה.
אוקיי, אז המתח של... אוקיי, סבבה. בלבלתי פה מלא מלכים, אבל... כאילו הבאזלר ממברנה זה אור התוף בעצם?
לא. לא, זה... תסתכלי על Auditory System, כאילו פלא כזה. הבאזלר ממברנה זה... אור התוף זה בעצם ממברנה מאוד פשוטה, שמחוברת לשלוש עצמות שבע. אור התוף נמצא באזור שיחסית האוזן התיכונה, זאת אומרת לא בהרכיב האוזן החיצונית, יש לנו את האוזן הפנימית שהיא נמצאת ממש בפנים. אור התוף בעצם קולט את הוויברציה, עוברות דרך 3 עצמות שהן הכי קטנות שיש לנו בגוף. שהעצמות האלה משנות שבעצם יוצרות כאילו ויברציות בסופו של דבר בממברנה בזית. למה?
אני רק שתולות בריאותיות זה לא סדר.
לא בריאותיות?
לא.
אין דבר כזה.
זה בדיוק, זה גם נותן.
זה לא בריאותיות.
לא מה אני מפורסם.
לא, זה אני אומרת, כי יש
לא.
משם.
יש כל מיני.
אבל אמרנו שהיא צריכה להיות גדולה, כאילו שאני לא קובלת את גוף. כדי שזה יספיק לכמה אנשים, הרי זה בדינה חוזרת מחרתיים. הסיכום שמי עד שהיא חוזרת, אז יש לך שבוע. ותכף יצאת בזול, כי אחרי החידת עושים פעלות מגיע לשם.
לא יכולו לאכול איתך את זה.
לא, זה לא תנאי. אתה יכול קשה?
זה היה בזה מה שאלה שבדינה לא תהיה, בדינה לא אוהבת. אתה זוכר?
אני לא יודע מאיפה הכנסת לעצמך את הדבר הזה. אני לא אמרתי את הדבר הזה. חוזרת לעצמך.
תשאל, בדינה, אני אכן...
את מייצרת לעצמך זיכרונות?
לא, ממש לא.
שלא קיימים.
לא נכון, לא נכון. בדינה גם זוכרת את זה. אפשר לדבר איתה על זה. ואת לא זוכרת שאמרת, זה בטח איזה אוכל שיבובי, זה משהו ממליז או משהו, וזה כל כך בטוח שאני רואה בזה. אתה לא זוכרת את השיחה הזאת, אנחנו דיברנו על זה. טוב, בדינה זה בדיוק ככה. רק שלא נראה לי שאפשר לאכול את זה. אותי אמרה. שאין מצב שזה שככה נגמר. אני יכולה להגיד את זה
מה קרה שאין בו?
לא, גילי הייתה אמורה, וגילי אהבה. לא, היא לא מרגישה את זה.
חוזרים ל8:30.
Okay, so I don't want to get into too much to the physiology of the auditory system in general. Sorry, but it's just important to understand that the fact that the memory potential of the hair cell itself, okay, the fact that it oscillates, determines the frequency in which the memory potential can oscillate, determines the frequency which the cell is tuned to, for reasons that I will not get into. And the component that determines the sensitivity or the frequency of the memory potential oscillation is through this potassium channel, and the fact that we have different splice variants just means that you can have in each, like I told you, that each hair cell expresses a specific combination of the isoforms, and thus you can see that through the specific combination or different combinations of isoforms of one gene, the cell can further tune its properties in order for it to be sensitive to a specific type of frequency. Okay? So, this from the book, that we know that there is at least one alternative splice site that is suppressed when a specific protein kinase is activated by depolarization. So, and actually we know that HNRNPs and SR proteins are extensively modified by phosphorylation. So this is like one of the mechanisms that we know that may cause alternative splicing, and may cause that you will have one isoform instead of the other, is determined, or the mechanism is facilitated through phosphorylation. Okay, so like I told you, like in the beginning, that calcium does almost everything in the cell. So also phosphorylation is a very, very important signal and modification that alters that happens inside the cell. And this is just to show you that the physical properties, like this is an analogy of a guitar, that you have the physical properties of the basilar membrane that determines like the frequency which it will vibrate in, but the alternative splicing is like the fine tuning of these strings. So, in general, when we're talking about alternative splicing, we know actually there's a lot of new researchers that are coming out on alternative splicing, and one of the reasons is that the techniques that we have today to identify different variants or different isoforms are much more advanced than what we had, like, let's say 10 years ago. And in the past, people have searched for differences or elements that may be unique that will determine like why are we different like from other organisms or from other animals. And for example, in 1964, it was thought that we have like that the human genome has like 1 million types of different genes, and this number kept on decreasing until today, and each one of these points is like an article or a study that thought that we have like 100,000 genes, and this thought we had 50,000 genes. But today we know that we probably only have like 22,000 genes. And does it sound a lot, 22,000 genes? So, for example, a grape has 30,000 genes, okay? A chicken has 18,000 genes, fruit fly 14,000, and then the influenza virus has like 11 genes. So, again, this is a note for like vegetarian people, but it has to be that the number of genes, it looks like that the number of genes has actually no significance, like, and if we're looking at like complexity or development or what is different between us and other organisms. And one of the things that people have noticed, that the percent of alternatively spliced exons in humans is the highest that was checked in general, and if we compare it like to a human or the mouse is very similar. If we look at primates, they're also somewhere between the human and the mouse. For example, there is a huge difference between the mouse and the rat in regard to the alternative spliced genes. And even more than that, when we look inside like the human body, and we look where do we have more alternative splicing, or where we have like a more diverse expression of isoforms, then the human brain is always the organ that comes up top, in the sense of it has a high percent of alternative spliced genes and variants. Okay? Yeah.
Before you said this, about 75% of the human genes are made alternatively.
Yeah.
And here it's still only to 25.
Yeah, that percent of the thing is that this is like a lot of times it's an average across tissues, and it doesn't like depict each identity of each gene. Like you can have in specific types of tissues, you can have just one isoforms, in other tissues you can have like multiple isoforms or other isoforms. So also here it doesn't mean like in the brain it's not necessarily the same alternative splice isoforms as in the kidney or the ovary, for example. Okay. So and also this number keeps increasing. So it's like what I showed you here, that the number of genes that we know that we have. I can show you another graph that shows you how many alternatively spliced genes we know that we have. Like we're always discovering like new isoforms of genes that were previously thought to have like less isoforms. Okay. So I'm not saying that this is what makes us like unique or the brain. There are other components, but it's still an interesting factor. So in respect to, I showed you here like changes that happen, you can say like naturally or affect alternative splicing or specific properties of specific genes. In this example, this is a research that was done in our lab more than 10 years ago by Mrna, which is today a professor here in the university. And what they actually found is that when they looked at the gene of acetylcholinesterase, they found that when, in this example it was mice, but when the mice were exposed to acute stress, then it caused an alternative splicing or an alternative splicing of the ACHE gene that, like to remind you that this gene is in charge of hydrolyzing or like terminating the cholinergic response, okay, the cholinergic transmission. Like I bring you back to when we talked about neurotransmitters, that the synaptic transmission normally stops by reuptake of the neurotransmitter, except from the case of acetylcholine, which is most of it is hydrolyzed by this enzyme. Okay, so they discovered that that not only that it changes, like the isoform or the splicing of the ACHE that is normally found changes from this S variant to this R variant. It's also not strictly like an axon that is added or subtracted, but there's actually like a new splice site in a location where there wasn't before, and what you get is like instead of exons 1, 2, 3, 4, and 6, you have this 1, 2, 3, 4 with an addition and exon 5. Okay, and what this actually alters, and this is again this is similar to what we saw in the article of Condell. This is in situ hybridization staining. Okay, so this is like staining against the RNA molecule, not protein. So they can see that in the hippocampal region you have an elevation, and this is a staining specific for this variant, for the R variant. So you have an elevation of this variant. And what's interesting about this variant in general, that you almost never see it. Okay, so it's very, very rare, and normally in like normal, if you don't do the animals anything, you can almost not detect this variant. Only when you expose the animals to acute stress, then you have an overexpression of this variant. And the mechanism that they and up until today, we actually don't know what is the mechanism that causes which causes this change or this alternative splicing. But we do have a nice movie that summarizes this, and it has music and everything, but it's very dramatic. Okay, so the components here are, these are splice factors, this is the ribosome, and this is the acetylcholine, which is also important to know that acetylcholine is mostly acetylcholinesterase, is in a tetrameric form. Okay, it has it has 4 subunits that are anchored to the cell membrane. So this is normal activity, and before you have like stressful conditions. And this is important, a question. Is this how the genome looks like inside the nucleus?
When it's not.
If the cell is going to divide.
If the cell is going to divide, but in this case it's not going to divide. So this is just like, you need to understand also the politics behind creating these videos, that you have to have like, this is what general people in the public are regular to seeing, like chromosomes, but this is not how chromosomes are spread. They're more like spread out throughout the nucleus. So what is this gene? This is RNA polymerase, and specifically RNA polymerase 2. We'll talk about RNA polymerase 3 in the... So what happens here is that this mRNA undergoes splicing. Obviously this is very, very simplified, but each one of these like cookies is a spliceosome. Okay, so what actually happens here? Why do we have this loop? I will repeat it like also you'll see it in following slides. It's very important because I think I told you in the previous classes that when a mRNA has to go translation, it forms this loop that has proteins that associate the 5' cap with the polyadenylated tail. So this is how you see it here, but without. And this is the protein. You see it's dramatic, and now it's being shipped to the synapse, where it can hydrolyze acetylcholine in the form of membrane-bound tetrameric. So in stressful conditions, it was normal.
Wasn't expanded when it's at the stressful condition.
It's just to emphasize that what happens to the protein, what's the difference between when you have normal conditions and stress. And I will talk about that in a second, okay? So here you have a lot of time when you have like serial stress, you have a lot of transcription also. And in this case, what happens is that you have alternative splicing, and the blue thing is cut out, and instead you have the red tail that you didn't have before, okay? So, what does this effect actually cause? What does it cause for the mature protein? It actually causes it not to be able to form these tetramers and not to bound to the synaptic cleft, okay? And we suspect that that this actually really lowers the ability of this protein to hydrolyze acetylcholine, because it also can diffuses outside of the membrane, and it's much less efficient in the hydrolysis of acetylcholine. So, it's also less stable. Okay, and I will stop here, and we'll talk about this second part later. But till this day, we don't know, like, if this is kind of a protection mechanism, or this is an how do you call it? Something that is bad, like what?
Feature.
What?
Backward feature.
Backward feature? What?
Feature.
Bug or feature. Okay. No, it's never a bug, but yeah, maybe it's like, does it create like problems, or is it help the system cope with stress? And I'll show you. We'll talk a little bit about that in the following slide. So, yeah. it's not the hydrolysis itself.
Any more acetylcholine in these animals that are stressed?
Yeah, there will be an elevation in acetylcholine in the synapse. There will be less efficient degradation of acetylcholine. Okay, because it has this alteration in the 3' end, and again, here it's not depicted, but it's a good lesson for you that a lot of times in an article they will not say which is the 5' end, which is the 3', but like I told you, this is the 5' end and this is the 3' end by consensus. And also here, because the exons are numbered, you can also know that the molecule starts from the 5' end and ends in the 3' end. So the alteration in the protein happens in the 3' end of the molecule. Okay? And it actually causes it to have like a tail that is unable to bind to the membrane and to associate with other acetylcholinesterase enzymes.
And also, what is the
The blue thing is an element that is normally not, it's not an axon, so it's like, it can be either, it's just here, it's to depict like an area of this axon over here that is also included in the coding region. Okay. It normally is not an axon here, but here it is an axon. Okay. It's just to show like that you have that you have different variants that you can have. Like the splicing can also, there are also variants that include like 1, 2, 3, 4, 5, and without the blue segment here. So it's just to show like another area that can undergo like different splicing. Okay, so question? Up until now we talked about this processing of eukaryotic mRNA, and now after the processing, then this mRNA has to be shipped outside from the nucleus and into the cytoplasm. And here I'll just mention like a very small point about the nuclear pore complex, which is a huge protein complex that is in charge of the transport of not only mRNAs, not only RNAs, but also proteins that tend to move inside and outside of the nucleus. And this is EM microscope images where you can see this basket formation that is in the nucleoplasm, and these filament-like formations that are in the side of the cytoplasm. Okay, and just to remind you that the nuclear membrane has is composed of two membranes and with a lumen between them. So this structure, do you remember that when you talked about the Candela article, we mentioned that when they wanted to overexpress or to express a certain gene, they introduced an intron inside that expression vector. Okay, and what I told you is that a lot of time there are many cellular mechanisms that are there to determine whether an mRNA was properly processed or not. Okay, and the thing is that after the processing, there are still, like I told you, there are still many proteins, like in this case RNP proteins, that are bound to regions where the splicing occurred, and this is actually like an indication or a tag that tells the cell this mRNA has been processed, and there are elements that know how to associate associate protein elements of this basket or this nuclear export. How do you call it? Nuclear pore complex, that associate the nuclear pore complex with the mRNA that is now exported from the nucleoplasm to the cytoplasm. So it was thought in the past, today people are not doing that so much anymore. It was thought in the past that you have to have like splicing of an mRNA if you want it to be like exported outside the nucleus and being translated. Okay. So, yeah, the fact that the mRNA was spliced leaves a mark on that mRNA, a mark in a sense in the form of specific proteins that are bound to this mRNA, and these proteins associate with elements inside the nuclear pore, which enable it or facilitate the export of that mRNA from the nucleus to the cytoplasm. Okay, so the last chapter of this chapter, I don't know what to call it, of this class is the cytoplasmatic mechanisms of post-transcriptional control. Yeah.
How, like you said something about the Candela article there, I don't understand. How was this not used
In the Candela article, it was used. Okay. The fact that they induced like this artificial splicing, which was not later expressed. Just a minute, I'll show you. I need to find it. Is this one? Yeah. Okay. So in the Candela article, we saw this example. This is the vector that they actually like introduced and caused the like expression of, and they included here. It doesn't look like that, but they included here an intron. Okay, so why to introduce like an intron in this element if later it will not have like an effect? Okay, so the reason that they introduced here this intron is that they thought in the past that these kind of elements help for this construct stability. Okay, so this can help like if mRNA undergoes splicing, then it also means that it will be like more stable or get exported properly, and vice versa. But today people are not using this. It depends. There are certain elements. A lot of time it's elements that cause like folding of the RNA that are regarding to like secondary structure of the RNA that increases stability of that protein. But I think that also I think that this was not proven like to be very efficient in general. But never mind. So where are we? Okay. Oh no, shit. And just to remind you the whole. So we talked about processing of eukaryotic pre-mRNA, and all these processes normally happen in the nucleus. It doesn't mean that there are no evidence that these processes can also happen outside the nucleus. Then we talked a little bit about macromolecular transport across the nuclear envelope. And now we want to know what are the changes that an mRNA can undergo after it's being exported and when it's in the cytoplasm. Okay, so the mechanisms that control or regulate mRNAs in the cytoplasm. And we'll start by introducing the notion of microRNAs. And microRNAs are small non-coding RNA, meaning that they don't have, their function is not through, they don't code to a protein, okay? Their function is through their form, is through an RNA form, and they repress the translation of specific mRNAs, and the processing is being one of the processing of this molecule, among others, is facilitated by an enzyme called dicer, that is a cytoplasmatic enzyme that cleaves this molecule and forms a single-stranded molecule, like it forms a double-stranded molecule that is later loaded to a complex that is called RISC, which is RNA-induced silencing complex. And through this complex, the mRNA, the microRNA, facilitates its function. Okay, so in microRNAs, they can be encoded either from inside introns of genes, or they can have their own promoter. Okay, so you can find microRNAs that This is one of the interesting phenomena a lot of times that you can find. You remember that, like, what happens to these introns when they're spliced out? So it's interesting to know. Like there are many microRNAs that are sitting inside introns of other genes. So part of the question is if they are transcribed as part of the large molecule, they have their own promoter inside the intron, but there also are microRNAs that have their own promoter outside the context of the gene. Okay, so in general, it's a question whether to call these genes or not, but we call them genes, or we can say that they're like small non-coding RNAs. And the function of the microRNA is through targeting of a specific mRNA according to the sequence of a 7 to 8 nucleotide sequence inside the microRNA molecule called a seed. Okay, so if we have our microRNA molecule that is normally 18 to 25 nucleotides in length, it has a seed region of 7 to 8 nucleotides that can bind to the target mRNA and recruit and cause actually the binding is done together with this RIS complex and can recruit or inhibit the translation of the target mRNA. Okay, so it can either, and also in some cases, it cause a cleavage of the target mRNA. So not only translation inhibition, but target mRNA cleavage. Okay.
What is cleavage?
Just cutting, like we said, endonuclease. It's like an endonuclease activity that cuts the molecule in the middle. So because the binding is facilitated just through these 7 to 8 nucleotide sequences, it actually it means that there are a lot of options for a single mRNA to bind to multiple single microRNA to bind to multiple targets, okay, or to multiple mRNAs. It doesn't mean that the same molecule will bind to a lot of targets, but the same, like the single microRNA kind or type of the same sequence can bind to many, many types, even thousands of different mRNAs, okay? Because the specificity, again, of a 7 to 8 nucleotide sequence is not is specific in one hand, but it's not very specific on the other hand.
When it binds to, like to a specific 7 to 8 nucleotide, does it suppress the entire gene that is like the entire mRNA that it bound to?
Yeah.
And also when it cleavages everything?
So when an mRNA is getting cleaved, normally the degradation of the rest of the mRNA is very fast. Okay? And also what I told you is that the microRNA actually interferes with the association of this loop that you have to have in order to initiate translation. So it actually suppresses the translation of the whole gene, of the whole mRNA. Another type of regulatory molecule.
Why does
Yeah.
this exist? I mean, why aren't these mRNAs just not transcripted or spliced differently?
First of all, I think we're gonna answer a little bit of this question in the following slides. But in general, the thing that we're discovering more and more and more, especially from our lab that we're studying RNA, is that there are always new layers and new layers of regulation. Okay, and if we have to say like what is the difference between higher vertebrates, like for humans and other organisms, a lot of times is in the complexity of the regulation. Okay, it's not that these actually microRNAs exist along like they were initially discovered in C. elegans. Okay, and they're also found in in low organisms as well. But the fact that they enable the cell to like silence the expression of a specific gene and not like completely lower or completely abolish the expression of that gene. So it not. you can transcribe it less, but then it will be, for example, it can take you like longer in order to if you want to make like a new mRNA, then this process can take longer. And also, like I told you, you can think of this whole system a lot of time. First of all, it's very complex, and it's very hard to speculate why the system has evolved as it did. But you'll see in a few slides that the regulation of microRNAs cannot, like the effect that you can have by regulation of microRNA is not the same effect as you can like start transcribing like a new mRNA or halting the expression or transcription of a new mRNA. Okay, so but obviously this is very, very complex, and we don't know a lot about it. Yeah.
The 7 to 8 nucleotides,
Mm-hmm.
is that in mRNA or is that
So in both. So there's a complementary sequence on the mRNA that the microRNA binds to through this element. Okay, so I'll show you. It's like if you have the mRNA that is going like here, then you'll have like a 7 nucleotide sequence that the microRNA will bind to. There's actually like more association in this area, but mainly the recognition is done through the seed sequence. Okay, another type of small RNA molecule, which is mainly found. It's not really found in higher organisms. It was mainly found in lower animals and discovered in plants. Is the siRNA, or Small Interference Rna, and these molecules are normally double-stranded RNA that are 21 to 22 nucleotides long, and they have, this is supposed to be complete, okay?
And microRNA 1 strand or 2 strand?
The microRNA, so the microRNA is 1 strand, okay, that is just like folding, it folds on itself, and then their enzyme called dicer like cleaves this area and part of this area, and creates like this double-stranded RNA, which is later loaded in a single-stranded form into an enzyme called AGO that is part of this RISC complex that I'll talk about in a second. So the siRNA starts, again, the function is through its single-stranded form, but it starts from a double-stranded RNA, okay? That is, and it's cleaved to the size of 21 to 22 nucleotides long, again from the same enzyme called dicer, like I showed you before. But this time it has complete compatibility with the target. Okay, so the siRNA, the main difference between microRNA and siRNA, apart from the fact that microRNA is like the natural, siRNA is not really found in high organisms and mammals, and microRNA is. So siRNA is like has complete compatibility with its target RNA, and the effect that it causes is not inhibition of translation, but cleavage of the target protein, of the target mRNA. Sorry. Okay, and like microRNA, it acts through the RISC complex, but this time the RISC complex induces cleavage and not silencing. Okay, so it has full complementarity with its target. So, in contrast to microRNA that can bind potentially to hundreds of targets, the siRNA is very specific. So if you have, for example, a 21 nucleotide sequence in the genome, it's very hard to, like in from probabilistic reasons, it's very hard to find a specific sequence of 21 to 22 nucleotides. So it's a very, very specific sequence. Okay, so
Why doesn't it in a higher or sentence weight? Because it makes more sense that higher organisms will need more regulation.
I don't know, but
Like you need much more molecules this way to do the regulation
yeah,
compared to mRNA.
it's true that you need to have very specific. You need to produce very specific molecules to inhibit specific genes. Okay, so a lot of times the siRNA is like in plants, it's made from the same gene that it inhibits. Okay, so these are two different small RNA molecules or non-coding RNA molecules, and they have also different effects. Yeah. In some
sense, that if the microRNA can do both, then you don't really need
yeah, but again, I would not. I would not speculate too much about why a system has these. It's just important to understand the differences between the molecules and their ability, or the processes that they facilitate. Okay, so if we're looking at microRNA precursor or a double-stranded RNA precursor, which in the case of siRNA, then these two molecules undergo cleavage by an enzyme called dicer. Then they're loaded on this RNA-induced silencing complex. And through this RNA-induced silencing complex, they can either, in the case of siRNA, have perfect base pairing with the target and cause cleavage, or in the case of microRNA, have incomplete base pairing. And this is because it's an old book, but in general, today we don't really think that that these base pairs are very important, but mostly we're focusing on the seed region over here, and in the seed region is actually what facilitates the binding between the microRNA and the target mRNA, and it causes an inhibition on the translation of that mRNA. Okay? So here's a chart that I'm not going to go over that, but it summarizes like the difference between these two molecules. And for example, microRNA today, they're really gaining significance also in brain science. Initially they were discovered in cancer, like in different types of cancer that it has been shown that they're upregulated or differentially expressed in states of cancer. But today, also in neurobiology, they're gaining a lot of interest in these microRNA molecules. The siRNA molecules are mainly used for experimental purposes. For example, if you want to silence or you want to downregulate a specific gene, then what you can do, you can just physically inject these siRNAs inside the cells that you want to target. And although we don't have these siRNAs in our body, we still have the mechanism through dicer and this RIS complex that can facilitate the action of this molecule and silence a specific type of gene. Okay, and they're also gaining a lot of interest as therapeutic agents. So I don't know if you heard about like gene therapy and all these there's a large debate about if we can, for example, if we know that a certain disease is caused by a mutated gene, for example, and you have let's say you have two alleles of that gene, or you have two copies of that gene, and and if you decrease the level of one of the variants, then you'll solve the disease. Then you can potentially just give a person these siRNA molecules, and it will specifically decrease that type of mRNA or that type of gene. Okay, so it's very interesting, and this you cannot do with microRNA. Like microRNA you're more worried about off-target effects because it can have these 7 to 8 nucleotides can bind to multiple targets. So the story is a lot more complicated. People are trying to pass drugs and patents and try to do it in humans, especially because you can engineer this molecule so it will be very stable, and then it will not contrast to normal RNA, it will not be sensitive for degradation. And then, and because it's very small, it can also undergo it can pass also membranes. So it's a interesting drug, a next generation drug that pharmaceutical companies are working on. When we're looking at the nervous system, and this time it's an example of two microRNA researches. And for example, like Cell is a very, very good journal for you to choose from an article. Also Neuron, which is from the same publishing group as Cell. And what the researchers showed here is that when they focused on a specific type of microRNA, in this case microRNA 182, they saw that in the retina of this, I think, was Drosophila, they showed that during dark adaptation, the levels of that microRNA decreased and then went back to the like steady state levels in light conditions. So do you remember that we talked about light adaptation in general, that we said there are like the phosphorylation of certain receptors helps for light adaptation, and also the abundancy of the specific protein along the cell, right? Do you remember that we saw that G protein couples, like the G proteins, move along the cell when we were in light conditions and dark conditions? So, for example, this can be another mechanism of adaptation. If you have a microRNA that will be transiently expressed or transiently reduced, then what it will cause is a release of inhibition of a specific set of genes. Okay, and in this way, it can be like a very simple, relatively simple solution of the cell if it wants to if it wants to overexpress, for example, a specific set of genes that are targeted by this microRNA. Okay, and they also found it for not only microRNA 182 but also other microRNAs. They found this phenomenon, and another paper. This time it's published in Neuron, showed actually the effects of overexpressing microRNA 125B or microRNA 132, which are both microRNAs that are known to be very enriched in the nervous system. Okay, and also microRNA 132 has been shown to modulate LTP, like processes of memory formation and LTP. And what they saw is actually that if you overexpress this microRNA, it causes a dramatic change in the synapse in the spine morphology. Okay, and I didn't show you like the normal spines, but this expressing this microRNA or expressing this microRNA, overexpressing this microRNA, caused like this opposite effect on the spine morphology. In this example, the spines were like elongated and thin, and in this case, the spines were like stubby and short. So obviously, the shape of the spines, did you learn about it, like about the shape of the spines and how it determines the current and stuff like that? No? So, what? So when we're looking at the spine of a neuron, the properties of the thickness of the neck, for example, determine how easily, for example, a current. Because what do you have in the synapse in general? You have entrance of ions or exit of ions. It doesn't matter, which is actually a current, right? So a current, the ability of a current to create voltage, for example, depends on the resistance of this component from the other components. For example, if we want to create like high voltage between these two regions, then we have to have high resistance between these two regions. The thickness of the neck of the spine really determines this factor and the effect of the synapse on the dendritic shaft that it's connected to. So the morphology here is very, very important for the sensing of the cell and the effect that activating these synapses will have on the current or the voltage that is induced inside the cell.
Maybe I missed it, but can these morphology changes explain the changes that we saw with the G protein, or those are just different mechanisms both responsible?
So they can. Like I don't know if someone looked at it. Actually.
Moving molecules or something.
So that's a question. Actually, I thought about it this morning when I went over the slides. I thought about maybe the possibility, because no one tells us if the actual proteins are moving or they're actually like not expressed, okay? But the thing is that my bet would be that it's probably both, okay? So you don't have only like protein translocation and movement, but you also have a local translation of proteins at in specific regions. And we'll just show you like a movie to summarize the concept of siRNA and microRNA, and then we'll take a break. Where is this? What did I do? Oh. Oh, you're not seeing it. This is also very dramatic music.
Scientists have been making rapid progress in understanding RNA interference, or Rnai. Many organisms use Rnai to control genes, and it can also be used as a tool in the laboratory.
Okay, just say, when they say RNAi, they mean either they mean like a group of interference RNA or small interference RNA, which includes inside itself the siRNA and microRNAs. Okay, so RNAi is like a general, like a popular name.
In the future, perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of cells, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II.
This is a more exact representation of how
The primary RNA
DNA
is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm.
You see the proteins are associated to the exons?
Ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. the targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaut catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide risk to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs.
They don't show it here, but actually what happens here is that the association, a lot of times this association of the risk complex, prevents this forming of the transcription loop. Just a minute, I think we're done.
Can you repeat what the transcription loop is? Like what are its ends? What is it defined?
We're going to talk about that in the next slide. Okay? So take a break and we'll come back in 15 minutes.
Does the siRNA bind strictly to only mRNAs that have 21 nucleotides, or can bind to longer mRNAs?
There usually no
They're
very short.
So the difference is like that. He said that it's like target specific just because 21 straight mRNAs is more specific. Okay.
It's very unlikely to have the same 21.
Since 1 to, I mean, since you have
4 to the
also
power of 20.